Good evening. Sorry it took so long to get this one out to you guys. Got a little distracted. I'm sure you understand how that goes. Okay, so as we talked about earlier in class, that there are lots of things that can go wrong with our three processes that we've learned about: DNA replication, transcription, and translation. Remember, replication is DNA to make more DNA. Transcription is DNA and make some RNA out of it. And translation is RNA to proteins. Okay, so there's lots of ways to mess up, and all the different ways you can mess up come in two varieties, either gene or chromosomal mutations. And so depending on how big the oops is will determine which one we follow. Okay, the first one is called a gene mutation. We call it a gene mutation because it usually affects only one gene. Makes sense. It involves DNA on the nucleotide level, so all the A's, T's, G's, and C's if we're talking anything involving DNA, and if we're talking RNA, it could be AU's, G's, and C's as well. So there's a couple of different kinds. The first one we call a single letter change. It's also referred to as a substitution. And so a substitution is just like me going, take that out and put a V in its place. So I'm messing with one letter, which is only affecting this one word, and all the rest stays exactly the same. So these ones usually aren't too bad. So this one, if it codes for the same amino acid, well then we're okay. If it codes for a different amino acid, well it could be either harmful or have no effect whatsoever. Every once in a while we do get some that have a good effect, but usually they have some bad. Okay, then we have a reverse order, which takes two letters and just kind of rearranges them like this. So bases turn into BSAES. And so again, it only affects this one word, and so therefore everybody else is fine, just this one is bad. And then we have a deletion, which takes out one letter, but then everybody else has to go down and fill in the blanks. So we have Feb and then aces. And so it wouldn't make any sense because this guy would go take in this place, the space would be here, and then this would be the word. And so as you can see, it affects everybody from that mutation forward. So deletions are pretty bad. And a deletion is also referred to as a frame shift mutation because it shifts all of the bases either up or down. Another type of frame shift would be an insertion, which is what happens when you shove a letter. Say I'm going to stick a Q in there somewhere. So I get the word addition, qual. Oh, wait, no, let me go back. Where'd it go? There it goes. Because the Q is now taking the last place, and so now the L becomes the first letter of the next word. And so now, as we can see, these words just don't make any sense. Same thing in our proteins, is that if our DNA gets one shoved in there and everybody scoots down, then the rest of the amino acids don't make any sense. So here's some examples. I was trying to show them to you with the words in the previous sentence, but you can see here. Here's our sentence. This is our DNA strand that we're trying to decode. And excuse me. And if I take out the I in time, replace it with an A, it becomes tame to dream, which still kind of makes sense, but it only messes up with one word or one amino acid, and everybody else is okay. Then we have reverse order, where I take the letters and flip them around, so time becomes emit, but to dream is fine, so only one word gets messed up, or one amino acid. Then we have a deletion, where I take out one or a couple of letters, and everybody scoots down to fill in the blanks. So here's our first word, second word, third word, and we can see that none of it makes any sense. So wherever the mutation occurs, everything afterwards gets affected. And then we have an insertion where I shove a letter in and everybody gets shifted down that way. And again, as you can see, everybody from that point forward gets messed up. Now, if any of these type occur at the end of the protein chain, well, who cares? One amino acid isn't going to make a difference most of the time. But if this occurs, 2 or 3 nucleotides in, well, then the whole entire protein is going to be completely messed up. Okay, second type of mutation is called a chromosomal mutation, which involves whole chunks of DNA. and all the genes on it, because a DNA strand is not just one gene, but it has several genes all along it, and every single one of these genes codes for something else. And so a chromosomal mutation may affect, this much of the chromosome, and therefore every single gene that is placed on it. The first example of a chromosomal mutation is a deletion, and just like it sounds, you remove a large chunk of DNA. So if here's my DNA, then what's going to end up happening is I'm going to remove a chunk and all the genes that go with it, and then I'm just going to take this part and scoot it over and glue it on here. And so, so now my chunk is shorter and it's missing a whole bunch of genes. Next kind is a duplication, where I'm going to take a chunk of DNA and duplicate it and then add it on, so my DNA actually gets a little bit longer, and it's got to repeat here and here. And so having more DNA is not necessarily a good thing. It usually causes problems. We have an inversion, where we have our chunk of DNA, and then this part, so pretend that's the middle, kind of rotates from here to here, and now this middle segment is completely backwards. Then we have a translocation, where we have two chunks of DNA that are right next to each other, and then his leg and his leg trade places with each other, so they each get a brand new leg. So a better way to explain this is by looking at this picture here. So we have a deletion. You can see that the gene for B is now missing. With duplication, the gene for B is double inside. We have an inversion, where A and F stay in their right place, but it gets broken, and B through E gets flip-flopped around the center. And then we have the two legs right here that break off and trade places, and so you can see where the two legs have been messed up. Okay, there's lots of different diseases caused by mutations. That's what a lot of doctors do, is to just identify these things. Here's a couple of them. Down syndrome is caused by having extra DNA. Cystic fibrosis is caused by one nucleotide put in the wrong spot, which creates the wrong amino acid, which causes lots of problems. Color blindness, like we looked at here, there's a number, I'll talk about that in a second. And then albinism, which is the inability to make melanin, so you have no pigmentation to you. Now, color blindness is a recessive disorder that you get from carrying it on your X chromosome. So guys tend to get it more often than girls, because they'll have it here on their X chromosome, and they only have one, so if you get the bad copy, well, you get it. Whereas girls, they have two X's, so if they have a good copy and a bad copy, well, that's okay. The good copy overrides, and so they're perfectly fine. So over here, like we saw in class, we've got a couple of tests. And so the first thing is to find the number inside. And so there's a number 6 right there. So if you can't see the 6, you might be red-green colorblind. Now this one, there is a 5 that goes right along here. But if you look very carefully, there is also a 2 that goes along there that's more pink. And then down here, depending on your ability, there's an 8 right smack in the middle. Or you might even just see a 3, because this little chunk right here is darker than these in here, and so you might only see the 3 instead of the 8. There's lots of tests that you could do online. There's websites that you could take these tests, and it'll tell you exactly what type of color blindness that you have. So you can check that out sometime, and tomorrow we'll go over everything. So remember, there's 5 sections, so there's going to be 5 quizzes, and all these need to be done by Thursday. All right, have a good evening. Sorry it took so long to get this up. Talk to you later. Bye-bye.